All right, is it visible?
Yes.
Yes.
Okay. So I start. Well, first of all, clearly, thank you very much for the opportunity to speak today. I'm presenting this work that Enzo has introduced, modeling immune recognition with restricted Boltzmann machines, which is work that I have done in collaboration with Simona Cocco, Rémi Monasson, Thierry Morin, and Alexandra Valsac at École Normale Supérieure in Paris, as well as Jérôme Toubiana, now at Tel Aviv University, and Vinod Balachandran and Ben Greenbaum at Memorial Sloan Kettering Cancer Center in New York. So let me first introduce a bit this process of immune recognition. So when the cell of an individual is infected by a virus, for example, this infected cell will start to display on its surface short protein portions that are called antigens and that carry the information that the agent causing the infection is foreign to the organism. Therefore, antigens can be then recognized by the host immune system. And in particular, here I will focus on the recognition by immune cells that are called killer T cells. An important question when studying these systems is actually what protein portions are antigens that can be recognized by the immune system. Because the antigens that can be recognized are clearly then important targets for the design of vaccines. And here I mean both the vaccines against a certain virus, because in the case of a viral infection, we know that viruses start to use the cell machinery to produce their own proteins, and therefore a detailed knowledge of what portions of these proteins are the viral antigens that can trigger an immune response is instrumental to the design of vaccines. But also, the identification of antigens is important in the context of immunotherapies that are increasingly acknowledged as a new pillar of cancer therapy. So a cancer tissue actually contains many mutations that give rise to mutated proteins that can be recognized as foreign by the immune system. And immunotherapies are actually broadly understood as a series of cancer treatments that are aimed at boosting the immune response of patients to cancer. And the success of this response depends on the recognition of these cancer antigens containing cancer-specific mutations. And so cancer antigens are now highly sought targets for the design of a type of immunotherapy that is called anti-cancer vaccines. But in terms of biophysical mechanisms, immune recognition relies on protein-protein binding processes, and in particular, there are two steps. So first, the antigen is displayed, so presented on the cell surface, and this is the result of the binding of the antigen to a surface protein that is called the human leukocyte antigen, HLA protein. And next, there is the proper recognition step that relies on the binding between the antigen and a surface protein of the T cell, that is the T cell receptor. And our objective in this work has been to actually build models of these protein-protein binding processes, really leveraging the information available in large-scale protein datasets. So first, let me give maybe a more general perspective on the approach we want to pursue. So, as we know, proteins are composed by amino acids, and antigens, as I said, are simply small protein fragments. So their amino acid composition can be actually visualized as simply a string of letters, where each letter stands for an amino acid. And thanks to high throughput sequencing technologies, we have access to these large data sets of antigen sequences that can be seen as statistical ensembles of proteins that share the same function. In this case, all antigens that are presented by a certain HLA protein. But similarly, also the T-cell receptors are proteins, and therefore the amino acid composition can be studied in terms of a sequence of letters. And again, high throughput sequencing gives a very detailed mapping of the individual repertoires of these immune receptors. And in these datasets, we can find a series of statistical patterns. That is, for example, the occurrence or the correlation in certain amino acids at certain positions. And these are actually the statistical patterns that reflect the constraints arising from the protein-protein binding processes that I have described. But since the collection of sequencing data is already massive and constantly fast-growing, on the other hand, data that describe these protein-protein binding processes at the level of structural resolution or measure of the binding energies clearly lag significantly behind the scale of sequencing. So an important question is actually how to extract from the sequence patterns information about the process. And to answer this question, the strategy is to solve an inverse problem. That is to try to infer from these sequence patterns probabilistic models describing these processes. And in particular, I will resort to statistical physics models that can well describe the observed statistics, but at the same time are amenable to an interpretation in terms of the mechanisms of binding. So then let me first start with the problem of modeling antigen presentation. That I said is the process by which an antigen is presented by this HLA protein. And what I should stress is that this protein is actually a very diverse protein. There exist many HLA protein types in the human population, and already the cell of an individual can display up to 6 different HLA proteins. So we start from the experimental data sets that sample the thousands of antigens that are presented by the cells, for example, in a tissue. Each antigen is specifically presented by one HLA protein type. And these records, therefore, provide sets of antigen sequences. And as I said, there will be some sequence statistics informative about the effective rules of presentation. That is, what combinations of amino acids are crucial to this binding process to the HLA protein. So the idea is to infer from the statistical patterns a probabilistic model, or in a statistical physics perspective, a complex energy landscape that effectively describes these rules. And the probability that is connected to this energy landscape will be then interpreted as a probability of presentation of a certain short protein. So to infer this landscape, I resorted, as the title said, to a machine learning scheme that had the restricted Boltzmann machines, here with the acronym RBM, that actually received attention quite recently, also in the context of protein biophysical modeling, thanks also to the work of my collaborators. So the model itself is defined by this two layers architecture. So it consists of a layer of so-called observed units that actually stand for the residues of the antigen sequences we want to model, connected to a layer of hidden units by a set of connections that are called weights. And mathematically, the model is specified by the joint probability between the observed sequences and the hidden units, that is written again as the exponential of some energy function, which makes therefore the model to be mapped onto a statistical physics model of interacting observed and hidden units. And from here, then, the probability distribution we are interested in, the one over the antigens, is recovered by integrating out the hidden units. But the advantage of having introduced these hidden units is in the fact that since they are connected to all the observed units, they enable the model to capture high-order correlations, therefore global patterns along the observed sequence. And the parameters that appear in this energy, that are, for example, these local fields on the observed units, these local so-called potentials on the hidden units, and the weights are not given a priori, but their values are inferred from the data in such a way as to best match the statistics. And this is done through a maximum likelihood type of learning. So we start from a sample that could be a custom sample consisting of antigen sequences, and we train, so we learn an RBM model of presentation by the HLA proteins present in this sample. And next, we validate the model's predictions on new sets of sequences. And in particular, the main model prediction is precisely the inferred probability of presentation. That is assigned to new sequences as a probabilistic score that is able to discriminate presented antigens from generic short proteins that are typically non-presentable because they do not have the right composition in terms of amino acids to bind to the presenting HLA proteins. And here I quantify actually this ability to discriminate presented antigens and generic proteins in terms of the probability that the model assigns higher scores to the presented antigens than to the generic ones. And as we see, the performance is excellent because this value is close to the maximal one. This type of model prediction is precisely the one which is useful in biomedical applications to identify what viral antigens or what cancer antigens are presented from the sequence alone, therefore potentially saving the time and resources needed for the experimental validation of presentation. And here I show that I assigned these probabilistic scores of presentation to sets of antigens that were detected in cancer samples, verifying that they would be correctly retrieved among the presentable sequences. Next, I want to recall your attention to the fact that we are modeling samples that collect together antigens that are presented by different HLA proteins. And in principle, the presenting HLA protein, what I will call the antigen HLA type, is not characterized a priori. And typically very few HLA annotations telling what is the HLA specificity of the single antigens can be retrieved only for a few of them from the existing databases. So a second important question is actually what are the presenting HLA proteins for given sets of antigens. And in this regard, what we find is that, again, the layer of hidden units plays an important role. That is the one of providing a space of representation for the data that enables dimensionality reduction, as I want now to illustrate. So we can define a projection of the observed sequences onto the set of weights entering a given hidden unit. What I call here the inputs to the hidden unit. And here I show the result of this projection for two hidden units chosen for illustration, where I show that antigens that have a different amino acid composition end up occupying different portions of this representation space. That is effectively the lower dimensional representation onto which the model maps the original sequences, and where a clear cluster structure emerges. What I find is that this cluster structure corresponds to antigens that are specifically presented by the different HLA proteins in one sample. Therefore, these different clusters have the interpretation of a different binding energy to the HLA proteins that clearly has a dependence on the particular amino acid composition of the antigens in the different clusters. So then the idea was to leverage this cluster structure to actually predict the antigen HLA type that I said is not always characterized a priori. And we do so by implementing a classification algorithm in this space that uses the information on a few antigens in each cluster to then predict the HLA type of all the other antigens in the same cluster. So let me summarize then what is the overall approach we are proposing. We start from samples of antigens that sample the antigens that are presented in by certain HLA proteins or in a certain tissue. And we learn from these samples a probabilistic model that is an RBM. And this model is learned in an unsupervised way, that is simply in such a way as to best reproduce the observed statistical properties of this initial sample. And this model has per se a value because it gives already a presentation model that can be used to assign probabilities of presentation in such a way as to identify what viral antigens or what cancer antigens are presented. Next, we can project the antigen sequences onto this space of input to hidden units. And here, antigens are partitioned into different clusters, as I said, depending on their amino acid composition that reflects their HLA specificity. And therefore, we exploit this structure for the prediction of the HLA type by a second machine learning architecture, so a simple classifier, that, as we see, connects values in this representation space to the actual HLA type. And the connections of this classifier are learned in a supervised way, that is, in such a way as to associate the values in this space to the HLA type, which is given, but only for a very small amount of antigens. In particular, what the results I will show are for a number of known HLA types that is equal to just 10% of the samples. And these HLA annotations can be taken from existing databases. So in this second step, that is the one in which the prediction, the model gives a prediction about the HLA type, what we are performing is a semi-supervised learning that is supervised at the level of the classifier, but only by a very minimal amount of known HLA types. And in particular, it is crucially guided by the cluster structure that has been already discovered by the RBM in an unsupervised way. And we propose this second part of the method as a tool to characterize especially newly available samples, starting from the very few HLA annotations that are already existing in the public databases. So here I want to show a benchmark of the prediction performance of this approach. And I do so by applying the method two sets of data for which we know the HLA type. So we can benchmark the performance precisely by comparison to this known HLA type, let's say the ground truth. And the performance here is defined in analogy to the discrimination performance I mentioned before. So it's the probability that the model assigns higher scores to the right HLA type than to the other types that are present in the same sample. And therefore it is maximal at 1. And here we see that the RBM actually reaches a very high classification performance, that is, it is able to classify the antigens by their HLA type, but also performance that is stable across the different datasets. And the other comparisons I show actually are meant to show that the cluster structure discovered by the RBM is crucial for this high predictive performance. So as I said, the approach we are pursuing consists of learning the RBM and then performing classification in this representation space by assigning a few types to antigens in each cluster. If the same classifier was applied directly in sequence space, so by assigning types to a few antigens directly in sequence space, the performance would be much lower.
Barbara, you have 4 more minutes. You have 4 more minutes.
4 minutes?
Yeah.
Okay.
Yeah, but 1 or 2 minutes more is no problem. We will cut a bit short on the question. So go ahead, sorry.
Sure. And the same conclusion can be drawn from here. So if we stick to this cluster structure, even without the classifier, and we just group antigens by a standard supervised clustering algorithm, then again the performance is rather high and much higher than if we clustered directly the sequences. So in a second project, then I tackled the problem of modeling actually the specificity of T-cell recognition. That, as I said at the beginning, consists of a specific binding between the surface receptor of the T-cell to the antigen. And to do so, I considered some datasets from our collaborators who are interested in detecting responses to cancer antigens by stimulating samples of the patient's T cells by these selected antigens. So upon this binding, a T cell becomes activated and starts to multiply many times, giving rise to population of T cells all with the same receptors. So in these experiments, after sequencing, we obtain samples that sample this recognition process. And in particular, these are samples of sequences, but this time from the T-cell receptors. And each sequence is present in a different number, with the highest numbers corresponding to the most expanded T-cell populations. So these numbers are informative about what T-cells responded. And the idea now is again to infer some landscape that would be in this case the landscape of T-cell recognition of a certain antigen, and I perform this inference by the RBM. So by these inferred probabilities, this time I can assign scores of specificity to the receptors that can discriminate the receptors that are specific to a certain antigen from the ones that are unspecific, that is, they recognize other antigens or they do not respond at all. And next I can again project onto the RBM representation space these responding receptors, precisely as I explained before, and this projection separates small clusters of receptors that share some sequence patterns that might be crucial for the binding at the molecular level, therefore providing precise input for additional validation of these sequence patterns. So I discussed a statistical physics inference approach to modeling the processes of antigen presentation and immune recognition based on the restricted Boltzmann machines. And I focused on 2 aspects. So first, these models allow us to assign probabilistic scores either scores of presentation to identify what cancer or what viral antigens are presented, and scores of specificity of T-cell recognition of certain antigens. And then I discussed this idea of projecting the data onto the RBM representation space, which is a way of discovering sequence features that are crucial for the interpretation of specificity of binding. And for example, in the case of antigen presentation, I have shown how this projection onto the representation space enables accurate predictions of the HLA type. And as avenues for future work, we would be interested in exploring the generative performance of these models. That is, by sampling from the inferred probability distributions, we could obtain antigens that are potentially functional, or receptors that potentially recognize a given antigen target. And these potentially functional sequences, of course, could be also tested in terms of their binding properties. And therefore, we think it is definitely an interesting direction to pursue. With this, I finish, and I thank you for your attention.
Okay, Barbara, thank you very much. I will clap myself. And
yeah.
Just representing all of audience. So, do you have any questions? Any question?
Well, I have a question.
Yeah. Please go ahead. Now I don't know, because I don't see the hands. I don't know what to do, but I mean, just one of you go ahead.
Okay, so let me, mine will be very quick.
Yeah.
Yes.
Okay.
So when you say that you are able to predict a new HLA, it means that you are able to design them with the specific properties. Is that the case?
You mean for the last point I mentioned?
Yes.
Yes. So the idea is that, for example, you can learn a model of presentation by a certain HLA protein. And then if you sample, for example, by Monte Carlo simulations from this inferred probability, then the idea is that you are sampling antigens that are presented by this HLA protein. So in this way, you would really propose sequences that are not seen in nature, but that potentially preserve precisely this function of being presented by a certain HLA.
Okay,
so yeah, these are sequences to propose for really design of new proteins.
Okay, thank you.
Thank you.
Any other question? Yeah, Mario.
I had another question. Hope not to be too long, but it's very, very nice, and your talk was so clear. So the fundamental question I have is whether you can use this type of methods in the classification you obtain also to learn the principles whereby those type of recognition are striving those recognition. And I guess you can do something like that.
Yeah.
And the other is more technical, and of course you had to skip on the details of the methods of the restricted Boltzmann machine that you use. But of course, I think all of us has in mind how the structure of that is affecting the type of imprints that you extract. I mean, for instance, I'm sure that the number of hidden units is going to play a role in the classification scheme that you get, and so on. Thank you so much.
Thank you for your questions. For the first question, absolutely, we can. The advantage of using a rather simple architecture as the Restricted Boltzmann machine is precisely also to give some insight. And I didn't show it explicitly, but the idea is that one can inspect what are the values of these learned weights that, for example, allow you to isolate in a certain regions the antigens that are specific to a certain HLA. And so you are somewhat highlighting what are the combinations of amino acids that are crucial to the binding to a certain HLA. So this is a type of insight we can get more about, let's say, the biophysics behind. And the second question regarding the structure of the model. So there are hyperparameters in the model that are, for example, the number of the hidden units, but also regularizations. And of course, these parameters must be set a priori by some cross-validation. So effectively, what I did was to monitor the overfitting with the number of hidden units, as well as the classification performance. And I could find that already with 5 hidden units, the classification performance was almost optimal and was kind of saturating. So it was rather stable from that point of view. But yes, the number of hidden units must be set based on this criteria.
Okay.
Thank you.
There are more questions? Very good. So if there are no more questions, I thank again Barbara.
Thank you.
And